before, so does anybody have questions before we get started on this? Or anything that's come up? I heard good things that I think one of you submitted a grant to the American Heart Association, I think it was, that Rob had mentioned. But if there's no particular questions, I can just kind of go through the presentation. And, you know, as usual, please interrupt me because I have absolutely no cues about what anybody's doing or anybody might have questions, okay? All right, so today's focus is on all the stuff that you have to do for a grant that isn't the research plan. And I title it Ignore the Rest of the Application at Your Peril, because I think this is probably the biggest mistake that I see young investigators make, in that they are so wrapped up in focusing on that research plan and that grant body, they forget the rest. And that you maybe can get away with it for some grant mechanisms, but for the NIH has criteria scores. And even though the research plan is the biggest driver of the score, 3 of the other criteria scores are not at all related to the research plan. So you really have to do a good job on the research plan, and being sloppy about it really does hurt people's priority scores. So that your grant application has some major non-research plan components, and I'm focusing this entirely on NIH, but other grant agencies also have the same requirements. And the reason the non-research plan components are so important is that they let the reviewer know that you can actually carry out the work. Now, how important and how much the reviewer will use those non-research plan components really kind of depends on what sort of privilege you're coming into the system with. If you're somebody who's like a really senior faculty member with a really, really well-known research program, you know, these components might not be as important because you have a reputation, so the reviewer is going to give you the benefit of the doubt because of that reputation. Similarly, if you're from a very, very big, extremely well-regarded research institution, everybody can accept that, let's say, Harvard or MIT has the research infrastructure necessary to get things done. However, that most of the people who will be listening to these slides, and definitely the 3 MRDA investigators, they're young investigators, so you don't have the reputation in your field so that everybody will just look at you and say, oh yeah, of course they can do the work. You know, they don't know you. They don't know if you can do the work. So you have to provide them the information necessary to tell them. And the other part is that about the environment, that we actually have some really, really great research infrastructure and resources in Delaware and in South Carolina. However, that it might not be out there with the national reputation. It's not somebody will say, oh, of course Nemours, or of course Christianicare, or of course MUSC or University of Delaware has what is needed to do a certain piece of work. Just because we have a lower research output than, you know, let's say a university in the Boston area or NYU or Johns Hopkins, or you can name better known research institutions. So the reviewers aren't going to give us the benefit of the doubt that we have everything we need to do our research. Again, then that means we have to tell them. So especially those of us, especially a young investigator applying out of an idea state, environment and Biosketch are really, really important components. Now, also we have budget and justification. For NIH, how much you have to put into that is a little hit and miss, because if you're applying for, you know, certain K awards or certain other smaller grants, the budget is, you know, for an R03, the budget is the budget. The justification, you're just saying, who do you going to have working for you to do the work? However, particularly if you're going for larger grants, and some clinical trials grants can actually have quite large budgets, you have to really justify what you need the money for and why do you need 4 postdocs and 3 clinical coordinators. If you don't justify why you need those things and you don't have that budget really, really solid, that they may give you a fundable score, but they're going to cut huge amounts of your budget out. So putting a lot of effort into that is important. And also the budget and the justification are what your grants offices are going to care the most about, because it's really a contract between your university or your hospital and the NIH when a grant is funded. And your organization really wants to know that you have the budget and the money you need to actually comply with the grant that you are saying and applying for. And then, of course, we also have the whole issues with research compliance. And these are having the correct animal and human subjects approvals, and not just the approvals, to have adequate information in the animal and human subject sessions. Also things that, do you have the biosafety components in place that you need? Do you have your data sharing plans? These are all things that take significant time to develop. And often people leave these for the last minute and do a bad job, and it really hurts their grant in the end. And one thing that I cannot emphasize enough, it is really important to work on those non-research plan components when formulating your specific aims. And you shouldn't leave it for last. There's this huge tendency to leave it for last because you're so worried about your grant body. I say, though, that you should be really doing that, start 2 months out at minimum. And I usually do this almost as a procrastination mechanism. Let's say I'm struggling trying to put together my aims or my research plan, and I have just burnt out my brain trying to write a paragraph justifying a certain experiment. That's the time to go work on the non-research plan components. That you can work on your environment sheet or make sure your biosketch is completely up to date. It doesn't take anywhere near the amount of cognitive effort. So you can work on these things when you're tired, particularly if you're doing it far enough out so that you can edit mistakes. But you don't want to leave them until the last minute. And the real big reason is that you will not feel like doing it near the deadline. Because when you're really making that last push to the deadline, you really want to focus on making sure that research plan is clean, is well understood, is in very, very good grammar, and that you don't want to, 2 days before your grant is due, have to worry about taking time to, work on your data sharing plan. You want to have that time that you can really, really refine the last minute changes to your research plan. But there's also other reasons of the importance of an early start, is that a number of you work not just within your institution, but you have collaborations outside of your institution. If you're doing community engaged research, you're doing collaborations with community health centers. If you are doing work maybe with a small college on an educational component, you can't assume that these external collaborators have all of the institutional compliance paperwork in place. And if, let's say, you're doing research with a community health center and they're receiving federal funds, they actually have to do all the institutional compliance paperwork. It's simple stuff like the DUNS number and all of the very other associated things. That paperwork can take months to complete in some cases, and you need to know whether or not you're going to have to work with those collaborators to get that in place. Another big thing to appreciate is that if you're collaborating with folks who do not apply for grants regularly, or they apply for grants regularly but not through the NIH, that it can take significant time to get Biosketches and environmental pages in a compliant format, particularly now with the new Biosketch formats. Is that if you have a collaborator who, let's say, is well-funded from the National Science Foundation or through PCORI, they're not going to have an NIH-compliant Biosketch. And now each Biosketch has to be customized to the grant mechanism and to the actual grant that's being applied to, because the biosketch is supposed to sell why that person is so great and why they're participating in the work. And you do not want to be chasing biosketches the week before the grant is due. That is extraordinarily stress-provoking. And you may not be able to get the chance to get it into the right format. And NIH kind of depends on what they're feeling like. I have definitely seen NIH grants get bounced because of formatting issues. And while I've also seen NIH grants go to review with formatting issues, you don't want all that hard work to basically be thrown out because you don't have everything in compliance. Another one is, again, if you're working with people who, in a collaborative setting, let's say you're working with a clinical collaborator, you cannot assume that that person has an ERA Commons ID, which is required for all investigators on an NIH project. And depending on your institution, it can take some time to get an ERA Commons ID. And if that's not set up right, you again do not want to be doing it at the last minute. Now, another one is IACUC and IRB approvals that NIH has this great mechanism called the JIT, or the just-in-time mechanism. But there are that where you don't need to show your IACUC or your IRB approval until in the months before the grant has been got a good score. That's when you need to worry about getting those in place. But there are a lot of other grant mechanisms that require these approvals at the time of application. And you have to remember that these are seldom quick. So you have to be aware that whether you need to have these in place or not. And then finally, use the staff at your research and compliance offices. That NIH particularly, they have standard deadlines. That regular research grants are always due June 5th, October 5th, February 5th, and and either competing renewals or revised applications are due the month later on the same dates. That if you can get all of the ancillary paperwork to your grants office significantly in before those deadlines, that will actually give the staff in your research office time to really go through all your documents, make sure they're all there, make sure they're in the correct format. However, if you send those stuff out a week before the deadline, everybody else's grants are also going in a week before the deadline. And that means the research staff just do not have the time to read all your documents and tell you whether or not they're in the right format or not. And that by that being the case, that can have, those kind of mistakes can result in a grant being desk rejected without ever being reviewed. So, the first thing absolutely is, work on all the ancillary paperwork, even while you're still doing the initial drafts of the research plan. Now, as you're going to try to really get that grant out, really think about, what are your steps? Very often people think of the first steps in planning a grant application is to write the grant. But really contact your department or your institutional grants administrators first. And most of the time, 2 months out is a recommended minimum to tell them that, yes, I'm submitting a grant for a June 5th deadline. That the reason is that, the grants administrators, they are trying to plan their vacations. That if most of the time grants administrators do plan their vacations around major grant deadlines. But let's say you're submitting to a different mechanism that's not around a major NIH deadline. What if that person who has to help you is planning a vacation or is going to be gone in that crunch period? This way you can get stuff done well in advance before they're gone so that you can get everything you need and you'll make your deadline and your grants people will know. And if you're working in a non-research intensive institution, you'll find the bureaucracy can often be tougher. And you might need to have even a longer lead time than just 2 months so that you can really use the grant or institutional grant administrators to help you. Because if you get close to the deadline, it's going to feel like they are in an adversarial position to you, because they're like telling you to rewrite stuff. They're telling you that, no, this can't go in this way. But their job is to make sure your grant is compliant with the rules and institutional policies. If you give them enough time, they'll help you get through this, but you have to give them enough time. And these grants administrators can help you understand what information you need to collect for the submission, both to comply with those grant and institutional rules, allows them to plan their workload, and also keep in mind that pretty much no research grants go directly to the investigator. These grants are made to institutions. And that, in fact, the official submission of the grant has to be made through the institutional grants office. So they have to be there to push the button to submit the grant. So under those circumstances, you really do have to be able to work effectively with their office. And they're helping a lot of people, not just you. So, you need to be sensitive to that in relation to their workload. Now, from a recommended timeline overall, 2 months out, compile a list of all the information needed. And you start collecting the information and writing the components. 1 month out, I would make sure you have all components drafted, and also you've made all requests for reagents, collaborators, and consultants. Because, very often, you need to get letters and biosketches from your collaborators. Those take time. Again, these are busy people. You can't expect them to respond at the last minute. Also, requests for reagents. If you need that mouse strain, if you need that chemical, if you need that data set, those requests and having letters certifying that those people are going to give you that information or those things can make or break a grant. And you don't want to have everything all drafted, counting on you getting a particular data set or piece of information. And then it turns out that the investigator who said orally they'd give it to you is out sick, or is at a conference, or has their own crunch deadline and they can't draft that for you. And that all necessary biosketches should be collected and editing for compliance begun. And 2 weeks out, that have all non-research plan components written and submitted to your research office for review. And that can even be further out, depending on whether this is an unfamiliar grant mechanism and also what your institutional rules are. Now, the advantage of having all of this done is that means that 2 weeks before that grant is due, you can completely and utterly focus on the research plan, and that all these other things which need to be worked on are things that you don't have to worry about. It's all done. All you have to do is focus on that research plan, which is all you're going to feel like doing in the 2 weeks before the grant's due. Now, I want to go through briefly just things that are some pretty important parts of the application. And a big one is the environment page, particularly for those of us who work in idea states. And again, this is because the reviewers are not going to give us the benefit of the doubt what our research environment is, what our clinical population is, and so forth. And the point of this is that it allows you to tell what the extent and quality of your dedicated research space is, as well as the dedicated research environment. That this is showing that your institution is interested in investing the resources in your success. That if you have a startup package as part of your faculty appointment, that should also be made very, very clear in your environment pages. And it should also be even made somewhat clear in your biosketch, because investigators that have the institution investing resources into them, that's saying that the institution is really invested in their success, and you're not just trying to do this as a side project. And that strong institutional backing is really important, especially if you're a new investigator. If you work at an institution that's unfamiliar to the reviewer, you have to write this specifically. I'll just use it as an example. The University of Delaware has a world-class bioimaging facility. We have as good or better microscopes and other sorts of imaging modalities than any institution in the United States. Now, that might not be something that's clear to somebody or somebody who would know that the University of Delaware has a world-class bioimaging facility. So it is, if you need to use that resource, you have to really, really sell that you have this important resource that you can do research that just on the surface nobody might expect. We have a world-class bioimaging facility. For those of you who are working on human clinical populations, you need to really explain and sell that you have the patient populations in the numbers and the composition that can really allow you to do this research better than anybody else ever could. That you want to, again, talk about your space, the composition of your department. What is your intellectual environment? Do you have people that you can just talk about to help you overcome research problems? What are your equipment, core facilities, and again, your patient populations? Now, for environment pages, if you have people who are working in a similar area, you can probably get somebody's canned boilerplate that kind of explains, let's say for the bioimaging facility, we have a boilerplate talking about all the great stuff that imaging facility has. If you have people who are working on the same patient population, let's say you're working through PedsNet or the really great medical resources available for pediatric research at Nemours, that somebody has written that up at your institution. However, make sure you don't just pop that canned boilerplate in, that you really want to read through it and you want to customize it to your needs. So you don't want to have tons of time talked about resources or things that are not relevant to your research project. And anything that is relevant to your research project, you probably want to elaborate so that there is very, very specific information that allows the reviewer to know that you're going to be successful. And do not include resources that are not relevant to your research plan. That just makes it really, really clear that it's boilerplate, and it really is a negative to the reviewer because they don't, like for instance in my case, they don't care that I might have access to a certain patient population if I'm not doing research with that patient population. Okay. Another one, of course, is Biosketches. Make sure that all of these submitted comply with the requested format. The current NIH one is common. Like this Duncan? Is something. Yeah, go ahead.
Hey, so it's Evan from MUSC. Sorry for interrupting. Mm-hmm. Quick question fine. About the environment page.
Uh-huh.
I totally understand the rationale for taking out parts that are irrelevant and then for deepening the discussion of parts that are relevant. For aspects that are appropriately worded, what amount of copy paste boilerplate is tolerated versus considered unoriginal, plagiarized from their perspective? Do they care that they're that's weird? This other guy from MUSC wrote the same sentence.
Yeah, I don't think that I've never heard anybody get called on plagiarism because you're using boilerplate environment pages.
Okay. It seemed reasonable to me that everyone did the same thing. I just was curious if anyone was that's weird. That guy wrote the same sentence.
Yeah, I mean, to be honest, there's this really funny thing that the University of Delaware Research Office makes us put at the end of our budget pages. That has it's basically legalese related to that how the budget process works. And at one point there was this thing of they made us put something about what is included in indirect costs, and they were and it was this whole big description of wafers.
Okay.
And they added this to my grant as boilerplate in the research office, and it's what the hell is a wafer? And it turns out buried somewhere in our indirect cost thing was something related to silicon wafers if you're doing synthesis of integrated circuits using silicon wafers. That if you need to buy the blanks for those wafers, UD can't charge indirects on that. But I do nothing related to that, right? So it just seemed weird. I ended up convincing them that it should be removed from my grants. But, I mean, there's just things like that. I don't think anybody's going to object that it's the institutional boilerplate that's used. I think more of the issue is that people tend to underwrite their environment pages.
Yes.
I've seen much more of a problem where it's like, oh yeah, I have 300 square feet, and I mean, that you have what you need to do the work, but this is a case of where you're not empathizing with who the reviewer is. You have a reviewer from Berkeley, and they know nothing about what happens at MUSC, right? They've never been there. They don't know people in your field from there. It just doesn't have a reputation in this particular area. And that it's your job to really, really explain that you can do this work. And actually, you might even be able to do it better at MUSC than at Berkeley. And that's actually kind of I think it's probably one of the biggest mistakes that I see. And some of it is that, you might have gotten somebody else's grant as an example, and it has very, very limited environment pages. Well, usually if you've gotten that grant, it's either from somebody who's at a better known institution than those of us who work in idea states, or it's a senior investigator. So, like, for instance, now I can probably get away with writing less of an environment page for one of my grants, just because, I've been doing this for 20 years as an independent investigator and I'm well known in my field. But that doesn't mean that a new assistant professor could get away with that, because that person hasn't shown that they can be successful in the current environment.
Okay.
That kind of makes sense?
Thanks for explaining.
And any other questions?
I have a follow-up question related to some of this boilerplate language, and this might be a little bit more for Erin, but are there resources available through the Excel website or other web pages where we can access some of the general language around research infrastructure at the Moores or the CPR? Is that available?
Hey, Nira, it's Erin. I know this is something Jim Sylvester was working on to get, the boilerplate for facilities and that kind of thing up and running. I don't know if it's been updated because, some of the labs moved to the experimental station and some of the labs are moving today to Rockland Center 1. So I can find out about that for you. In terms of the mentoring resources, a lot of folks have just used the Excel website and the GIN toolkit and have found that they can kind of take some of the language from there and use those to talk about the different mentoring resources we have.
And also take a look to see who else is in your institution who's doing similar stuff. I don't work on the clinical things you guys do, but here, for instance, there's a set of animal resource pages and bioimaging pages and stuff like that we all of us who write NIH grants, it's oh, how are you writing this section now? And they send us the text and we use pieces of it and we modify others for our own things. I know, for instance, the bioimaging facility, the bioinformatics facility, they have resource page pieces that can be popped in. And this is something, it sounds like from Erin's answer, this is some stuff that might be developing for some of what the clinical side is. But, for instance, if you're using PedsNet, somebody should have written a boilerplate for the access to the medical records of Nemours, which is a fantastic research resource, and to explain it and talk about its depth and all of that.
Right.
And that's actually, to Erin, that could be a really good resource for, the Nemours clinicians in particular.
Yeah, and
Okay.
we've talked about it. It's it's definitely also a little bit challenging because there's so many different types of labs, and then there's the standard stuff, but then there's different labs and clinicians. But two things, Nira, is we can always help you with any of that, so we could find something for you. So if you need it, let us know. The other thing is,
Okay.
as Melinda has said, work with our OSP office because they're seeing the submissions that are going in, so they would know. Who's using what language and how often is it used, and can you use some of it? So don't be afraid to work with them and find out who's putting in something similar to what you're doing, because if anybody knows, it's actually them.
Okay.
And this is also another reason where high lead time can be really, really great, because then you can, send the email and get the response in 5 days. And if you're working on this stuff 2 months out, then you can find and collect, the information that you need from the people you need when they're not time stressed.
Right.
Any other questions for right now? Or we can go on? Okay. So I'm going to go on to Biosketches. That this is a case, make sure all of these submitted comply with the requested format. And one of the reasons that I kind of emphasize this particularly to this group is that, I have seen like all the MRDA applications over the past several years. And very often, these are community engaged research people, or they're working with clinicians. And very, very often, the people you're collaborating with don't have NIH format Biosketches. They either have the old format of NIH Biosketch, which they update, or they've just never made one at all. And it is absolutely not a fast process to put together an NIH Biosketch right now. Some of this is because of that the first paragraph is basically customizes and makes the case of why somebody's contribution to this project, what are they bringing to it? What kinds of skills, what kinds of resources, what kinds of knowledge are they bringing to the collaboration? And even just writing that intro paragraph can take time, and it takes some thought to do it well. There's also the contributions to science section, is that you need to pick out the top contributions, and you have to write a small little paragraph saying what is that contribution with supporting references. And this can be hard to write simply because a lot of us are acculturated to not toot our own horn. We basically say our accomplishments should speak for themselves. But you have to talk about not just that you did certain things and published certain things, but this is why anybody cares about it. And this is really a marketing document. You're marketing yourself. That if you're an older scientist, you should only be probably mentioning your contributions in response to your publications. However, if you're an early career scientist, you absolutely can include unpublished contributions or contributions that are not traditional. And these can include manuals for things, standard operating procedures, GenBank accession numbers. You want to sell that you actually did contribute something important. You could put abstracts. You want to say, that you've done science and it's not just, it's important science. And it's one of the things for your own Biosketch, that's something you have control over. But you want to make sure that all submitted Biosketches, and this includes the Biosketches from your collaborators, your consultants, your mentors, comply with the NIH Biosketch format, or the format that is wanted for the mechanism, and that they're customized for the project. Like they can't just send you the Biosketch they submitted with their last grant. it has to be rewritten so that it focuses on your project and why your project is important. When the biosketch is clearly from somebody's regular grant and isn't from this project, makes it really, people start to wonder whether that collaboration or that mentoring interaction is real. Because it takes time to rewrite that intro paragraph, and that by customizing it, it shows that the collaboration is real and the collaborator is engaged. If it's just, anybody can take 2 seconds and send a biosketch from some other project that isn't related to yours. But this also means that you need crucial to give adequate time to get these appropriately edited. And the reason that I'm really emphasizing this is that many of the CTR pilot and MRDA applications, the collaborators, the clinical collaborators, and often the mentors' biosketches are not in NIH format. If you are submitting to any external thing, it's really, really important that those biosketches are compliant. So like for a mentor, it should say something like, I have the expertise to mentor so-and-so's project because I have done all of these things or have access to these data sets or whatever. It should really, really sell why they should be the mentor. Or if it's a collaborator, they have to really, really sell what their contributions can be to this specific research project. And not just, again, a biosketch that's clearly from another application. Now, a couple things that I would absolutely say also is that a couple things that aren't really in the format but can be really helpful. You want to list your total number of publications on your biosketch. And it might not hurt to say this both in the personal statement where I'm qualified to do this because I've published 20 papers in this area. And also in the contributions to science sections, like if you have published 5 papers on a certain topic, say that in the 5 publications that I have published on this topic, I have discovered blah, 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 blah. This is really helpful because that you may be well published or famous in a particular area of science, but the study section member might not know who you are. That I actually have a friend of mine who developed a particular technique. And while he was a graduate student, when he was applying for his first R01 application, that he used that technique extensively in his application. And he got one of the reviews came back and said that that there's no indication that this investigator knows how to do this technique, and that he needs to find a collaborator to make it convincing that he could do this technique. Well, he's the person who developed it, but his biosketch didn't, and other aspects of the application didn't make it clear that he would that clear straight out, it wouldn't have been an issue. And it was just, the person who was reviewing it wasn't aware that he was the key person who developed that particular technique. And that's showing, it's a really good example of showing that need to be able to communicate very, very clearly you have the skills necessary to do the project. And you cannot assume the person reading the application is going to know that. Another thing that can take a lot of time to put together is what's called the My Bibliography website. That NIH Biosketches now have the ability to link the biosketch to something called My Bibliography. This is set up on the National Library of Medicine website, and that there's a mechanism where you can add this URL to your biosketch. And that gives you the opportunity to put your actual publication record on your biosketch. You can't list it on your biosketch itself. And this is actually, to be honest, when I review grants, the first thing I do is I click on the My Bibliography for somebody that I don't know. And this is somebody's entire curated list of publications. This confirms actual productivity, which is important because the contributions to science narratives, everybody can tell a good story about what they did, but that URL should have your complete publication list. It should also have the complete publication list of all the collaborators on their biosketches. And this is a place where, especially, let's say you have a clinical collaborator who doesn't really apply to the NIH, so he doesn't have an NIH biosketch. He's published, 75 papers. That putting together my bibliography on their biosketch can take a lot of time, because that my bibliography site, you can start with a PubMed search, but if somebody has a common name, you have to remove everything that's not relevant. You have to add everything that is relevant, and this can be really hard to get done if you have a very busy clinical collaborator. This was kind of a nightmare for my clinical collaborator in Ohio, getting him to take the time to do this. He's very, very well funded. He's got tons of industry money doing research on various kinds of techniques. He has an enormous publication record. And when they had the new NIH biosketch, getting him to start his My Bibliography and get this biosketch together, even with a 2-month lead time, we almost missed the grant submission, because getting him to take that time out of his busy clinical schedule to get that done was really painful. Now, one thing about when you're doing your biosketch is that there's a rule that says you shouldn't place in preparation or submitted papers on the biosketch, because, and reviewers really don't like it, because especially in preparation papers are often fantasy. So it makes it like an attempt to inflate your progress. However, there's a new policy where NIH says that you can list intermediate research products. And these are preprints that are posted to preprint servers. So what this means is that if you have a draft paper or a submitted paper, you could upload it to one of the preprint servers, like bioRviv and things like that. And you can list those on your biosketches as intermediate research products. Nobody really knows how this is going to be looked at by reviewers or what it's going to look like, but it is a place that if you have a submitted paper that you want to have considered on your biosketch, you can get it on your biosketch by uploading it to a preprint server. All right, and then finally is the research support session. The research, for somebody who's been doing this a long time like me, you basically put all your grant funding from the past 3 years and you're done. However, if you are newly independent and you received startup funds or you have institutional resources to do your research, absolutely put that under your other support on your biosketch. And you can also talk about your dedicated research space or any other kind of special research support you could be getting. Let's say like a what's the word I want? A release from clinical responsibilities, because these kinds of things are in fact showing the institution is investing in you to get your research up and going. Of course, you list any current funding because this speaks to productivity. You have to include all funding you've received within the past 3 years. However, sometimes people will include older grants if there's space to show that you do have a publication record or a funding record. Okay. All right. So any questions on biosketches before I go on? Okay. All right. So the budget and its justification. This is the piece that your institutional research offices will absolutely care enough about. This is the listing of how much money you're asking for, along with the specifics of why you need it. Now, if you're going on an NIH modular application, these are reasonably painless. Your only thing you really need to specifically justify is your staff and their duties and percent effort. Those are for the modular applications. If you're applying for a non-modular NIH grant mechanism, as well as most other grant mechanisms, you have to explicitly justify all your costs. Now, this is a piece that the institutional grants officers look at really closely, because that if the grant is funded, it really is a contract between the institution and the funding agency. And they want to make sure that you've asked for all the indirect costs that you're supposed to ask for. Have you, like in my world, we have to ask for a certain percentage of graduate student tuition. They want to make sure you've done that and that you have asked for funding for all the stuff that needs to be funded. And generally, reviewers are asked whether your budget's reasonable. So you have to make sure that you've justified that appropriately. However, one thing that especially young investigators often think that might be a good idea is to lowball their budgets. So they think, well, if I really need $200,000, but if I ask for 125, they're going to look at this being a good value, and they'll be more likely to fund me because I'm not asking for as much budget. However, this can backfire big time. One is that if you have clearly lowballed your budget, the reviewers will lose confidence in your ability to do the work because you appear naive. It's just like, really? You can really get this done for that amount of money? I couldn't do it for that amount of money. So you really, and then there's the concern is that if you don't have the project can be great, but if you don't have enough money to do the project, then you're going to get a lower score simply because the impact of the project on science is going to be small because you don't have enough resources to actually do the project. Now, so part of your thing is to make sure that you do your budget justification so it's not lowballed, so the reviewer's convinced that you know what you're doing, you know how much it's going to cost, but you haven't asked for crazy amounts of money, so the reviewer's like, yeah, this person's just trying to milk the system. Now, keep in mind that even if the reviewer recommends what's called time and amount, in other words, the yearly budget and the number of years you're asking for, NIH can still cut the budget as recommended for time and amount. And they still somewhat expect you to do the proposed research. Just as an example, my competing renewal, the reviewers recommended it for time and amount. However, while they gave me the amount, in other words, they gave me the yearly budget, they only gave me 4 of the 5 years I asked for. Now, NIH will never increase a budget. So if the reviewer says, well, they need more money than this to do the work, NIH is never, ever going to increase the budget for a lowballed budget. So the most you can get is what you ask for. You can definitely get less than that. So take home, do not lowball your budgets. Really ask for a reasonable amount of money that you need to get the work done. Okay, any questions about budgets? All right, now human subjects. There's 2 aspects of human subjects that are involved in being a grant. One is the regulatory stuff, the stuff about your grant going through IRB and your project going through IRB and this sort of thing. Now, generally, NIH has this mechanism called just-in-time, where they don't ask for the final IRB approval until you've already been reviewed and they're considering your grant for funding. However, there are human subject sections in the grant. And for clinical research, this can absolutely make or break your grant. That they are really looking for strong evidence that you have considered fully the challenges of identifying sufficient numbers of subjects to get statistically relevant data, and that you've overcome these challenges. You have to be very, very specific about whether your subject population has that appropriate race, age, gender diversity, and you have to fully justify why the subjects do not represent the full diversity of the population. Don't worry about being too obvious. It's completely reasonable. If you're doing prostate cancer, just state why you have not included women or children. if you're doing Alzheimer's, state why you did not include children. That you want to make sure that you've given power analyses to show that your study population is big enough to get the desired, to basically test the hypothesis with a certain predicted effect size. That particularly if you're working on a reasonably rare disease, I'll use some of the really fascinating work done at Nemours on pediatric bone diseases, that the reviewer may not know that Nemours is the main clinical catchment for a certain bone disease. So, and they know that this disease is extraordinarily rare. You need to say that, Nemours sees X patients per year with that disease, that based on prior work, we anticipate a certain X percentage will be consentable for whatever is being done with that subject population. This consentable number is sufficient to be able to test our hypothesis based on this power analysis. These are things that are really, really important. And when I've reviewed grants, this is the biggest reason that grants using human subjects are rejected, is because this patient recruitment and power analysis stuff just hasn't been done well or clearly. Now, from the other side is that your research might be considered exempt or even not human. So not human research can have it could be that it's all cadaver material, that's all de-identified. That's not human. It can be from previously established cell lines, again, not human. I just had a research project declared not human yesterday, which was an interview project asking physicians about their opinions based on a certain kind of clinical intervention in a disease. That also was determined to be not human because it's not asking anything about the doctors, it's just asking their professional opinions. But even in those cases, you have to write the human subjects section. Like in my case, I would have to write, this is how I'm getting the doctors, this is how I'm identifying the doctors. I would have to state explicitly that my university's IRB has determined this is not human under this definition of human subjects research. If you're doing research that's exempt, you want to describe everything about your study population and then explain that it's exempt from IRB guidelines under this particular category of exemption for this reason, and that's been certified by your IRB. You don't want to have your grant reviewers flagging your grant for not complying with human subjects rules. And you will find that your grant reviewers are not well familiar with the extent of the exemption rules or even with the extent of what is human subjects research or not human research. You could be using, waste surgical tissue that's de-identified. That is non-human research, but your grant reviewer may not know that. So you should spell it out and explain why it's exempt or not human under those circumstances. These are things that could absolutely kill a grant if it's not done right. Similarly, I'm not going to put a lot of time into animal subjects because I don't think anybody on the call does do animal work, but it's the similar type of thing. And one really big new one is that, traditionally a lot of animal research was only done with males because males have less variability because they don't undergo a menstrual cycle or an estrous cycle. NIH now requires that animals of both sex be used in studies, and that this has to be explicitly talked about, that you're using animals of both sexes. You have to really justify all of your reason you're using animals in the first place. How are you minimizing pain and distress, and how are you undergoing euthanasia? Again, you do not want to be flagged by a reviewer for an animal subjects violation, and you need to put in the time and effort necessary to explain it. Now, one place, we were talking about boilerplate earlier, and absolutely you can use boilerplate for animal and human subjects sections. However, be extraordinarily careful about using boilerplate. Even be careful about using boilerplate from within your own lab or your own grant writing, because it's really easy for somebody to say, oh, I used animals in this grant, and I have my animal boilerplate from the last grant I submitted, copy, paste, done. However, that the reviewer can tell if they're clearly lifted from another application if you're not careful. For animal work, let's say you're studying a certain mouse mutant in a previous project, and if you don't fix the boilerplate for the new mouse mutant, it's like, no, you explained about how you're going to work with this mouse mutant, or you explained about a particular surgical procedure that is not used in the body of the grant. That can really mess you up. In a human subject situation, the human subject section is written about this human population, but it's a different study, so you're talking about consenting for a different procedure. That if those sections are lifted from another project, that can absolutely get you flagged, and it can really delay or even prevent you from getting the award. Also, again, don't be sloppy or vague. This is a really big problem where people wait to do these sections at the last minute. And they're like, oh, yeah, animal section, yes, I will lift it from my previous grant. Of the times that I have done grant review, you might get 10 or 11 grants to review. I've seen these mistakes usually on a good 10 or 20% of grants that I review, and it absolutely hurts your grant in the end. All right. Now, I've been kind of alluding to this just-in-time information thing. And just-in-time is that NIH does not typically ask for the final IRB approval number, the final IACUC approval number, until at the time of submission. They wait to ask until the grant has received a good score. And that kind of means that you're kind of tempted to wait to do your IACUC or your IRB approvals until you know you'll get a grant. However, this can be really dangerous. IACUCs and IRBs, especially if you don't have one in place right now, they can be really, really slow. They can also be kind of picky about whether or not they like the way you wrote your IACUC or your IRB protocols. And it can take months to get these things approved. Like even just my recent determination that something wasn't even human, it took me 3 weeks to get that done by our research compliance office. And it wasn't that, once they actually looked at it, it only took them a few hours, but they're busy. They have lots and lots of things on their plate, and it just hadn't gotten to the top of their list. And finally, I sent them a quick email saying, I need this for a particular purpose. Then they did it really quickly, but it sat there until I pestered them. So what I would say to you is that if you don't have your IRB or your IACUC approved at the time of submission, go ahead and start the approval process right after you submit your grant, because it might take you that 6 months to get your IRB approved. But if you have the approval at the time of submission, go ahead and include the number. Other things that just in time is other support and pending applications. This is an audit NIH does to make sure you're not double dipping funds, so that if you have submitted the same proposal to 3 different agencies, you're only allowed to take money to do the project from one of the 3 agencies. Now, this information, NIH will automatically request this information if your grant is in the top 30% of scores. But generally, go ahead and wait to do the just-in-time until they actually ask you for it in person by email, because often they're only going to fund the top 15% of of applications. So, there's no point in doing just in time for your 27th percentile grant unless they tell you to. Now, one thing that I just ran into, I just went through the just in time process with my competing renewal, and I found that this process has gotten a lot more complicated, and they're asking for a lot more information that they've ever asked before. One thing that you have to understand is you cannot submit just-in-time information. That has to be done through your research office. NIH will not accept just-in-time information from you directly. It has to go through your grants officer. And this means is that, when you get just-in-time inquiries, you have to respond very, very quickly. That some things that I ran into is that there's the just-in-time that's available on the website, but then when NIH starts to go through your application and that website stuff, they staff has a name similar to somebody who has been debarred from all federal funding, and that I actually had to go through the process where we had to have a certification process that the person on my project with that similar name was not the same person who was debarred. That held up the funding on my grant significantly, even though I was on it right away, just because it takes time to write this and write the certifications and get the research office to submit it. And then you go to the bottom of the funding pile. They asked me for the full approved animal subjects protocol, not just the numbers that it was approved, but my actual full approved animal protocol. Again, that also delayed things a couple weeks. So again, just keep in mind that once you've done well and gotten a good score, you have to still go through the just-in-time process and definitely get on it. I know people who have lost their funding because they did not do the just-in-time information in the time frame it needed to be done. Okay. And just a few other things that if they're appropriate to you, ignore at your peril because they'll flag your grant. One is the resource plan. So this is if you're making antibodies, if you're making genetically modified animals, if you're collecting a lot of data, particularly large-scale data from human subjects, there's a rule that you must share those created resources and make that data publicly available. Now, you probably can find a boilerplate for this about the legal aspects of data and resource transfer. It is okay to restrict such transfers only to qualified individuals. And your institution probably has boilerplate for that, but you should definitely customize that to your project. That a lot of times people don't put in this section and it gets their grant flagged and then it causes a giant nightmare later. They're not really that hard to write. They're usually a paragraph or less, but you do need to make sure they're there. You just want to have the reviewer say data and resource sharing plans are in good shape. A similar one is biosafety, is that a lot of applicants will ignore this. That if you're working at biosafety level 2, which are things like retroviral vectors, lentiviruses, viral infection vectors, sometimes even human clinical material, the section is required and you need to be writing this up. So, don't forget that if that's appropriate for you, you definitely need to take it into account. So with that, I think that's all I have to say to you about other grant sections. But, I think in the end, take-home message is just make sure that you, do these things, do them far in advance. I actually really do use them when I'm procrastinating about my brain being tired working on my research plan. I just pop these things out. I have a folder with it. I have a folder for the grant I'm writing, and I have a folder that's called, that's called like other forms or something. And if I really don't want to read any more literature, I pop out the research sharing plan, or I edit and update my environment page. So it's not painful, but you definitely do not want to have to work on it the week out. With that, I'll open it up for any other questions. But any other questions for folks? Back to Blue Jeans and see. I see everybody's still on, but anything else you want to talk about?
Do you have, I mean, just I know we're kind of short on time here, but do you have a sort of a recommendation for like order of approaching these things? Like what to tackle first?
I don't think it actually matters. I think that the first thing is getting a list of everything you need. And just realize that some of it's going to take you more lead time than others. So things like getting your letters of support and making sure like your clinical collaborator's Biosketch is good, I would probably prioritize that because that's largely out of your control. While something like writing your biosafety plan, that's something that, one day that you're working on your grant, you don't want to read another paper, just work on that, right? So I would definitely prioritize the things where you need more lead time.
Okay.
But I mean, and also you can talk to your research office because, there's certain things like the research office in our institution, what they really, 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 really care about is the budget. So, by getting that done and like to even get the process started in my institution, you need to have the abstract of the grant done, which is like the lay abstract, the public abstract of the grant done, and you need your budget. And if you get that uploaded, then you can start the paperwork approvals, which at my institution is about 8 layers of different people, everything from you to your department chair to everything else. You can get that process started so that you can get the institutional approvals you need to even submit the grant. So that's a reason to do that reasonably far ahead.
Okay.
But
thank you.
But there was another question. But anybody else? Well, like I said, I'm always willing to take email questions if anything comes out. But and Erin, for those of you who are at Nemours, Erin can definitely help with that. Trying to get this information together. For those of you at MUSC, I am sure that there are grants officers and stuff who'd be happy to help you with this. You just have to give them the time that you need to do so. Okay. So with that, I will let everybody go, and I will go on my way to the periodontist. Thank you very much.
Thank you.
Okay, so